Just give me one
So
second.
she's on.
Okay, just give me 1 second to get rid of this.
I think we will start.
Mhm. Okay, good to go.
Can you hear me? Yes. Yeah, okay. So welcome everybody in person and also virtually. Wow. Okay. So in case you don't know me, my name is Guanming Wu. I'm a faculty member in DMICE. I am quiz mentor during past several years. So I'm going to introduce quiz. So it is my great pleasure to introduce quiz here. And Quiz grew up in Buffalo, New York. So he got his bachelor degree in biology from American University in Washington, DC in 2016. He then worked in Thermo Fisher Scientific as a scientist, working on cell culture related projects. But he did not tell me, but I guess the life in a company at that time was not challenging enough. So then he decided to move from East Coast to West Coast here at OHSU to pursue a PhD degree in biology, bioinformatics and computational biology. So that was 2018, so a year just before pandemic. So fast forward, now somehow 6 years passed. That's a very, very long journey and adventure. So it is a journey to show Chris hardworking and tenacious, and it is also an adventure full of unexpected challenges and accelerated moments. Chris faces each obstacle with determination, showcasing his resilience. The adventure is not just about the past 6 years, but also about personal growth, self-discovery, and the pursuit of scientific innovation. So it is my true honor and privilege to have a chance to work with Chris during past several years. So Chris and I met almost twice per week. Of course, most of our topic is about science, about his research project, sometimes also my research project. But once in a while, we also talk about something else, like sci-fi stuff, like sci-fi movies, TV shows, and books. So very recently, we talked about a new Netflix sci-fi TV show called Three-Body Problem, and it's related to books. As you may know, in physics, the three-body problem is not solvable, but it may be handled somehow by using numeric approaches. So Chris' dissertation topic is about the cancer immune microenvironment, which contains much, much more than three bodies, and therefore probably in theory, much more complicated than the three-body problem. So could we do something also using computational biology for this specific problem? So let's hear what he's going to talk. So here, without further ado, let's see what he's going to talk.
Thank you, Roman. Now try to live up to that intro. Today, I'm going to be talking about some of my doctoral research that I've done over the past few years, and where I developed some computational tools to kind of unlock the ability to ask new questions of single-cell gene expression data. I then kind of applied some of these tools to certain questions relating to certain immunotherapy treatments and trying to figure out why some patients respond and why some patients do not. I want to start with the kind of high-level clinical motivation for this work. As we all know, the kind of burden of morbidity and mortality from cancer is quite significant, both in the United States and globally. And, many of these deaths from cancer come from patients who are treated with some of the standard treatment methods that do not turn out to be successful. So when we think of the kind of standard methods of treating cancer, we can think of surgery, chemotherapy, and radiation therapy. However, over the past several decades, we've seen the development of two new classes of therapies, which include targeted therapies and cancer immunotherapies. And the idea behind these immunotherapies is to harness the patient's own immune system in order to ideally target the tumor cells in a more specific way and leaving the surrounding healthy tissue relatively unharmed. So we can think of the human immune system as being a distributed system for the determination of self versus non-self. So we have various cells in the adaptive immune system, including the cytotoxic CD8 T cells. These cells patrol the cells of the body, and if they find a healthy cell, then they need to leave it alone. Whereas if they find a cell that does not belong there, either because it is infected with bacteria or virus, or because it is developing potentially into a tumor, it needs to identify that as something that does not belong and eliminate it. So in the functional case, these cytotoxic T cells can identify tumor cells by having receptors that bind to tumor antigens, which are mutated proteins presented on the surface of these tumor cells. At which point they can recognize them as not self and eliminate them. In the kind of healthy functional case, we have these CD8 T cells, which through this receptor binding can identify these tumor cells and kind of implement the body's kind of inbuilt anti-tumor immunosurveillance process. However, there are signaling proteins known as immune checkpoints, which can come to be expressed on the surface of these CD8 T cells when they are continually exposed to the same antigen over a long period of time. Then the tumor cells can express a kind of corresponding protein that can interact with this and perform inhibitory signaling, which can then diminish or turn off this anti-tumor immune response. And this kind of raises the natural question, at least for me, why would these CD8 T cells have essentially an off switch that can be hijacked by the tumor cells to evade this anti-tumor immunity? And this gets to kind of the careful balance between autoimmunity versus kind of effective targeting of pathogens and cancer. And so this is potentially an evolved mechanism to kind of downregulate this excessive immune response that might be causing damage to healthy tissue. But it can be exploited by these tumor cells when they are learning ways to evade the immune system. But this raises an opportunity for a novel class of cancer treatments known as immune checkpoint inhibitors. These immune checkpoint inhibitors are biologic drugs that take the form of monoclonal antibodies. And these antibodies can bind to the immune checkpoint proteins, interfering with this inhibitory signaling, and in the ideal case when they work, restoring this anti-tumor activity. So the kind of early success of these treatments in a subset of patients has kind of created a lot of excitement and attention on these therapies. For some patients who are treated with these immunotherapies as a last resort, after they've been treated with some of the standard methods which have failed, some of these patients can receive, they can benefit from years of disease-free survival, and this is really exciting. However, the reality is that many patients do not get this level of clinical benefit. A lot of patients show resistance, and you can't just give these therapies to everyone because number one, when you're kind of tinkering with this delicate balance of tumor targeting versus autoimmunity, you can get pretty nasty immune side effects like cytokine release syndrome, which can be very serious for some people, and they're also extremely expensive. So there's a need to kind of, number one, identify the subset of patients that's actually likely to benefit from these treatments, but then also to kind of identify the mechanisms by which other patients are not responding to potentially be able to expand the proportion of patients that are actually able to receive a benefit. So just to kind of compare this to the kind of normal functional case, we have typically a naive T cell, which will be exposed to its matching antigen, whether that's from tumor or some pathogen. It will activate and have a cytotoxic effect, ideally eliminating the source of that pathogen, and then a subset of those cells can be retained in a memory population in case of exposure to that same antigen later on. However, if for one reason or another these T cells are not able to fully eliminate the source of that antigen, either because it's a tumor that is not able to be fully eliminated, or because in the case of chronic infection, which is another context in which this can pop up, these cells will start to express immune checkpoint proteins and then be kind of progressively pushed along a series of cell states, starting with a more stem-like progenitor-exhausted population, and moving then on to a more terminally exhausted CD8 T cell population. And the relative balance of these two cell types has been implicated in how well a patient will respond to immunotherapy. So that raises kind of the high-level question for this work. What is the relationship between this process by which CD8 T cells move through this series of dysfunctional states and whether or not a patient is actually going to benefit from immunotherapy? Because these CD8 T cells are important in the mechanism of how these therapies work and are the target of the attempt to reinitiate this anti-tumor effect. But before I talk more about kind of the balance between those two states, I want to talk a little bit about this problem of heterogeneity. So when we're looking at a tumor, we have a mixture of different cell types, different immune cell types, subpopulations of tumor cells, all mixed together. And if we use classic bulk gene expression assays to try to quantify this, that signal is all averaged together in a way that it can be useful for some applications, but can make it challenging to answer more specific questions. And this is particularly important in cancer, where you can have subpopulations of tumor cells that, if resistant to your treatment, the treatment will essentially act as a selective pressure, leaving only those resistant cells, which can then proliferate and drive disease progression. So one way we can start to address this heterogeneity problem is by using single-cell gene expression assays. These are data where we have an expression profile showing the abundance of each gene's expression in each individual cell in potentially tens of thousands of cells within a sample. So this can give us a much clearer picture of what's going on in the tumor. And we can take this high-level data, use dimensionality reduction to get a two-dimensional plot like this one. And we can see here that we have these points which represent cells. And these cells that are closer together have more broadly similar expression profiles. We can then cluster these groups of cells into cell types, and then from there even further down into individual cell states. And this can give us a lot more information. So moving from our high-level question down to a more specific question about the relationship between T-cell exhaustion and immunotherapy, I started out trying to get a clearer picture of how the other immune cell types in the tumor immune microenvironment relate to these states and the relative balance between this more stem-like progenitor-exhausted CD8 T cell state and the more terminally exhausted populations. it bears mentioning that these progenitor-exhausted CD8 T cells and terminally exhausted CD8 T cells are not non-functional. They're merely dysfunctional, but they do have some anti-tumor effector activity. So the progenitors can proliferate into downstream cells, which can have an anti-tumor effect, and the terminally exhausted CD8 T cells can have an anti-tumor effect as well, but it's just reduced relative to what you would expect in a typical activated CD8 T cell. So we can use pretty standard out-of-the-box single-cell RNA-seq analysis methods to look at these two populations of cells, get a trajectory from one to the other, look at how gene expression and regulation kind of changes as we move from one to the next. And that's great. And it can give us a lot of insights into what's going on here. But a question that's a little bit more difficult to ask is how do other immune cell types in the tumor immune microenvironment relate to this process? So the kind of moving from the clinical to the biological to the more methodological motivation for this work, I wanted to come up with a way to be able to consider what's going on in these other immune cell types that could potentially have an important relationship with the relative balance of these two immune cell populations. So to do that, I developed a framework. But before I get into that, I'm just going to give a brief note on the data we're going to be looking at here. These are single-cell RNA-seq data sets derived from patient tumor samples with melanoma and basal cell carcinoma, which are two somewhat different skin tumor types. And we also kind of remembering that this process can occur in chronic viral infection as well, we want to kind of compare the T cell exhaustion process in the tumor case to the chronic infection case, so that we can kind of get a better sense of which findings from the chronic infection case actually generalize and are relevant to the tumor case and potentially immunotherapy response, and which are not. So to do this, I developed the following computational framework. We start with single-cell RNA sequencing data. We subset down to the cell state, the cell type of interest where we're going to be looking at this cell state change. So we, in this case, subset down to the CD8 T cells, and then find a trajectory, which is essentially a path starting from this TCF7 high, tox low, more stem-like progenitor exhausted population, moving progressively through to this more terminal population. And again, this is all analysis that we can do with pretty standard, straightforward tools. However, in order to be able to consider what is going on potentially with these other immune cell types, I had to kind of come up with a way to make this comparison. So in order to do this, we come up with a sample score. And this is kind of the crux of how we're able to make this comparison here. So we have this linear trajectory going from one cell state to the next, in this case, the progenitors to the terminally exhausted cells. And the cells that make up this trajectory are not all from one patient. They're from a number of different patients with a number of different tumor immune microenvironments. And the idea here is that the CD8 T cells from a given patient are not necessarily going to be distributed along this trajectory in the same way as other patients. We can have some patients like sample 1 in green whose CD8 T cells are kind of biased toward the earlier part of this trajectory, whereas other patients like sample 2 in red are biased more toward the later end of this trajectory. And what we then do is use a chaos test-based kind of statistical method to come up with a single score for each patient to quantify kind of the overall level of CD8 T cell exhaustion in that patient based on the distribution of their CD8 T cells along this trajectory. And what this allows us to do is, if you look at the bottom left here, we'll be able to identify patients like patient 1 in blue that is biased toward this progenitor exhausted state and compare it to patients like patient 3 in green, which is much more biased towards this terminal exhausted state. And then once we have these kind of broadly high exhaustion and low exhaustion sample groups, that allows us to make different comparisons. So we can compare the difference in gene expression and regulation between those sample groups in the cell type of interest itself, so in this case within the CD8 T cells, but we can also then move that comparison over to other immune cell types as well. So we can consider, for example, in the macrophages, how do the low exhaustion and high exhaustion derived cells vary kind of in different ways, and then look at some of the gene regulation patterns there. So after developing this framework, I applied it first to the skin tumor data. So these are on top CD8 T cells from our melanoma samples. You can see in the bottom, we start with a TCF7 high, tox low, progenitor exhausted state on the bottom. And as we move up into the left, you can see an increase in tox, a decrease in TCF7, and the upregulation of the immune checkpoints PD-1, LAG3, and TIGIT. So then we can take that linearly defined trajectory, you can map it onto the y-axis of the violin plot on the bottom, and we can see these are ranked from left to right by their sample scores, and we can see patients like the two on the far left side that have broadly low exhaustion scores and are biased towards this progenitor exhausted state, while we have samples on the right that are comparatively pushed much further along this trajectory. So then we can take the cells from these sample groups in the CD8s and then in other cell types and start to make our comparisons. Next, we can see this applied to the chronic viral infection data. So these are T cells from patients with chronic HIV infection. We can see the occurrence of these two same CD8 T cell states. And here, we can very clearly see that the samples on the right are much more biased towards this kind of progenitor exhausted state, while those on the right are biased towards the terminally exhausted state. So once the kind of high and low exhaustion sample groups have been identified, we can then start to make the comparisons. First, we can make the comparisons in the CD8 T cells. This is transcription factor activity that is either up or down regulated in the high exhaustion derived CD8 T cells relative to low. And this is activity that generalizes across these two different skin tumor types of melanoma and basal cell carcinoma. So the top line here is, kind of a nice internal kind of check that we're picking up on some of the same biological processes across these data sets. But what's more interesting is here with the macrophages, we can see that there are a number of transcription factors that consistently across these skin tumor environments are upregulated in the macrophages from the high exhaustion microenvironments. We can also look at this from a pathway perspective. So we can look at the change in some of these reactome pathways and see that in the macrophages from high exhaustion microenvironments, we see an upregulation of a number of pathways, including MAP kinase and various cytokines, including interleukin-10. These are pathways that are all upregulated across all three data sets. So I identified some that were tumor specific, some that were viral specific, and this is pathway activity that generalizes across the tumor and viral contexts. We can then use various techniques to start to look at kind of gene regulation between some of these transcription factors and pull out some of these modules. I found that NF-kappa B as well as STAT was consistently upregulated in the macrophages across all three data sets. So in this way, by applying this kind of sample score framework, we were able to make these comparisons and start to find some differences between these macrophages defined from different immune microenvironments. which brings us back to the original question. Are there differences in the immune microenvironment between samples that vary in their distribution of these states? Using this framework, it was able to identify the upregulation of NF-kappa B, STAT, and some other genes and transcription factors in these high exhaustion macrophages. So this gives a little bit more clarity of what's going on in the tumor immune microenvironments of these patients based on kind of their overall degree of T cell exhaustion. But this still leaves kind of a number of unanswered questions. So the focus on the immune microenvironment and the T cells themselves is important, but another kind of important piece here that I wanted to take into account is the tumor cells themselves. And these tumor cells are heterogeneous. We see different subpopulations. And the other thing I wanted to add here is the ability to make a more direct comparison to the kind of clinical question that drove this in the first place, which is, are these patients actually receiving a benefit from immunotherapy? So I want to bring in these clinical phenotypes of clinical outcome more directly. Which brings us to the second question and the second approach. We can see we have multiple subpopulations of tumor cells, and these tumor cell subpopulations can vary in how they relate to the immune system in kind of two broad categories of ways. So the first kind of most direct way these tumor cell populations can differ in how they relate to the immune system is just how likely they are to be identified and targeted by the immune system. So we can have one tumor cell population that might show downregulation of MHC and have reduced antigen presentation and maybe be less likely to be targeted by the immune system in this way. The other way we could expect these tumor cells to relate to the immune system is we might see differences in tumor subpopulations in the signals they release to the immune system, which may modulate the behavior of some of these immune cells and affect their ability to target not just that tumor subpopulation, but the entire tumor more broadly. So in order to answer some of these questions about tumor cell subpopulations and immunotherapy response, I started with the framework developed in part one and expanded it in order to be able to kind of incorporate some other information here. The two inputs here are single cell and bulk data, which kind of allows us to take advantage of the advantages of each. The advantages of single cell data are, relative to bulk data are pretty apparent. It's far higher resolution. You have a lot more kind of insight into the individual subpopulations of cells. But bulk data actually does have its advantages. It's been around for longer and it's cheaper. So often you can get bulk data that has higher sample size and sometimes is more likely to be actually labeled with your clinical phenotype of interest. So by kind of leveraging both of these, we can take advantages of the strengths of each in order to kind of support the analysis. The inputs to this are going to be pre-treatment single-cell RNA sequencing data from human tumor samples. So the exact same kind of general type of data as part one, except in this case, it's going to be from patients with non-small cell lung cancer. The bulk data is of the same tumor type, also pre-immunotherapy treatment, but is labeled with how these patients then went on to respond to immunotherapy. So in this way, we can bring in these phenotype labels. This approach has two kind of arms that are conducted in parallel and then brought together at the end in order to make a comparison. So we start with the same framework used for part one, selecting out, in this case, again, the CD8 T cells, getting the exhaustion trajectory and calculating sample scores in order to select subsets of lung tumor cells that are from exhaustion high or exhaustion low environments. And then in the second part, we can bring in some of these phenotype labels with the bulk data. But before I get into that, I just want to make a note about some of these clinical labels that I'll be using here. So these bulk data samples are labeled with immunotherapy outcome. And anytime that physicians are trying to quantify how well a patient is responding to a particular cancer treatment, there are a number of different ways to do this, but it typically starts with some sort of a scan. It can be a PET scan or PET CT scan. In a PET scan, you introduce radioactively labeled sugar into the patient's system, and since the tumor cells preferentially uptake sugar and have a high metabolic rate, these will kind of show up on the scan, and you'll be able to identify kind of primary and metastatic lesions. And then you can do these sequentially, and you can take a look to see if you see identifiable changes before and after therapy. So in order to quantify this, one way is using the RECIST framework, where any patient who has all of their lesions disappear after treatment is designated as having complete response. Any patient who has a significant reduction in the size of these lesions as no new lesions is quantified as or classified as a partial responder. Patients who have new lesions or have lesions that are increasing in size are considered to have progressive disease. And this is the worst outcome. And patients who are not, who do not clearly meet the criteria of partial response or progressive disease are considered to have stable disease. However, for the purposes of this analysis, I'm trying to kind of enrich for the most clearly defined responder versus non-responder phenotypes. So to kind of increase the statistical power to select the particular subsets of lung tumor cells we're looking for. So in order to do this, I set aside the stable disease patients and considered any patient who had complete response or partial response to be a responder, and any patient who had progressive disease to be a non-responder. And in this way, we had a binary clinical phenotype, which we could use to select subpopulations of lung tumor cells. Now, in order to do this, I made use of a tool that was developed by another researcher at OHSU, Zheng Xia at the Knight Cancer Institute, where we can use our clinically labeled bulk RNA sequencing data to select subpopulations of the single cells, basically by choosing single cells that have expression profiles that much more broadly match the expression profiles of bulk samples with one clinical phenotype relative to the other. And in this way, we can select subpopulations of cells associated most significantly with treatment resistance or treatment response, and then leaving the rest as a background. So I applied this tool in the second part of the framework here in order to identify these subpopulations of lung tumor cells. And then the final step of the framework is to compare the exhaustion-associated lung tumor cells with the phenotype-associated lung tumor cells. After developing this framework, I then applied it to these 3 non-small cell lung cancer data sets. We have 8 patients for which we have a good number of CD8 T cells and tumor cells. And then the Liu et al. Data set in green, you can see we have just a huge number of T cells and not many tumor cells, but these were essentially just used to help more clearly define this trajectory of CD8 T cells. On the right side, you can see we were able to identify CD8 T cells and tumor cells. These were subsetted out and then fed into each part of this analysis framework. So the CD8 T cells into the sample scoring framework, and the tumor cells into the phenotype association step. When we look at the CD8 T cells from the lung tumor samples, we can see a pretty similar pattern to what we saw earlier. So there are TCF7 high tox low cells on the right side, indicating our progenitor exhausted state, which move to more of a terminally exhausted state with the upregulation of these immune checkpoints as you move to the left. We can see that trajectory on the y-axis on the bottom, and we can see that patients on the left side have low exhaustion scores and are comparatively more biased towards this progenitor exhausted state, while the sample on the right is made up almost entirely of terminally exhausted CD8 T cells. So once these sample groups are identified, can then do downstream analysis in the lung tumor cells. So these are lung tumor cells where we have, on the left side, are phenotype associated cells. So in blue, these are lung tumor cells that are most strongly associated with immunotherapy resistance, while in orange, we have lung tumor cells that are most strongly associated with immunotherapy response. On the right side, we see the cells based on the exhaustion score of the microenvironment they're derived from. And what you'll notice here is that the lung tumor cells that are most strongly associated with immunotherapy resistance show significant overlap with lung tumor cells derived from these exhaustion low tumor immune microenvironments with a bias more towards this progenitor exhausted state. And then on the other side, you'll see the lung tumor cells most associated with immunotherapy response have the most overlap with the exhaustion high microenvironments. And this is a little bit unexpected based on what I find in the literature. Another way of looking at this is that we have our samples ranked from low exhaustion score to high along the x-axis. And you'll notice that the low exhaustion samples on the left side have a significantly higher proportion of these resistance-associated cells. And then the pattern shifts where we see more of an enrichment of the response-associated cells as you move to the right on the exhaustion high side. So another way to look at this is by performing a differential expression analysis between the low and high exhaustion groups in the lung tumor cells and between the resistance and response associated lung tumor cells as well. And we can notice that there are a number of genes whose activity is very significantly upregulated in both the immunotherapy response and exhaustion high groups. We see interferon signaling, including ISG15, which is also known to be able to drive IL-10 signaling. If you remember from part one, we saw an upregulation of IL-10 signaling in the macrophages from the high exhaustion microenvironments as well, as well as some STAT signaling. We can also look at this from a pathway perspective, again using reactome pathways. This is again in the lung tumor cells associated with immunotherapy response and high exhaustion microenvironment. And we can see more STAT signaling, which is driven by ALK, which is a gene that is commonly mutated in non-small cell lung cancer. And we can also see the potential tumor suppressor RhoBTB2. On the other side, there are a number of genes associated with immunotherapy resistance and an exhaustion low microenvironment. So here we see TIMP1, which promotes cell proliferation, various immunoregulatory proteins, as well as heat shock protein binding protein 1, which is known to be associated with poor outcome. So I developed this framework in order to bring in some phenotype information and identify subpopulations of lung tumor cells that are associated with a certain degree of exhaustion and a certain level of immunotherapy responsiveness. We were able to find some kind of unexpected patterns here. And in order to see how relevant these findings were potentially to other tumor types that are also candidates for immunotherapy, I took some data from melanoma patients, again, that also had phenotype labels, and derived a immunotherapy response signature from the lung tumor cells from the highly response-associated versus resistance-associated groups, and then applied this gene signature to the melanoma data and saw quite a degree of overlap here, which is nice because it suggests that there's enough shared biology between the kind of immune activity in these different tumor microenvironments that findings in terms of mechanisms of response to immunotherapy that are discovered in one tumor type that is a candidate for immunotherapy may generalize to other tumor types that are a candidate as well. So I've spent a lot of this time talking about cancer and immunology, but this framework really can be applied to the same class of question from a methods perspective across a wide range of biological and clinical problems. So, we can look at T-cell exhaustion, we can look at immunotherapy response, but we could also choose as a cell state potentially differentiation level of the tumor cells themselves, looking at tumor grade, and then look at a phenotypic outcome like overall survival. We could also step away from cancer entirely and look at questions in development about the degree of differentiation of a particular cell type during development, and then its association with a particular phenotype. So, these tools, I applied them to my kind of biological and clinical interest, which is cancer immunology, but they can be applied to a number of questions that are more broad than that. So I started out with some questions about the relationship between T-cell exhaustion and cancer immunotherapy response. This is a big field, so I can't say I answered all of them, but I developed the sample score framework in order to get a little bit of a clearer picture about what's going on in the tumor immune microenvironment that could potentially have an effect on this balance of progenitor to terminally exhausted T-cell states. And then to tie this more directly to subpopulations of tumor cells, as well as the actual clinical outcome of interest, I expanded this framework, allowing us to make more direct comparisons between this cell state change process of interest and a more clinical level phenotype. And with that, I would like to thank my advisor, Dr. Guanming Wu, as well as the rest of my committee, who has given me a huge amount of support and guidance along the way. The Wu Lab, our department, all my fellow students, and families and friends. Thank you.
Question?
Mike. Yeah.
Thanks, Chris. That was a really beautiful talk. It was nice to see you progress through, and I enjoyed looking at all of your slides. My question is related to the data sets that you chose and thinking about your findings, because I also was unexpected to see that the responders had the T-cell exhaustion high versus the other way around. So in your lung data set, Do you think that's being confounded in some way by like the timing of when the patient samples were collected, and were any of them receiving treatment prior to collection? Can you talk a little bit more about the structure of the data set and where the patient data?
Yeah, that's a good question. So these were all patients who had samples collected prior to immunotherapy, but these patients have all had previous therapy. As far as the what my thoughts are about the kind of explanation for that unexpected direction, I've been thinking about that a lot, and it's obviously the first kind of potential failure point is just kind of the setup of my framework. But I've taken enough steps to kind of increase confidence in the phenotype selection as well as the kind of sample score setup that I'm fairly confident about that. I think a more likely explanation might lie in the complexity of the T cell biology, and whether these are T cells that have been recently exposed to antigen and might express some of the same genes that you would expect in a terminally exhausted population. I've looked at, I've tried to dig a little bit deeper into that to see, if we can look at gene marks that differentiate between terminally exhausted and kind of just activated T cells that would differ enough from the progenitors to set up that trajectory. And it's not slam dunk conclusive, but the evidence points in the direction that they're probably terminally exhausted, not recently activated by my evaluation. So yeah, I don't have a super clear answer to that question, but it is unexpected, and I'm as I'm kind of writing this up for publication, I'm trying to figure that out myself. But yeah, it was certainly unexpected.
You'll get more from me, but what happens if you just change the direction of your trajectory?
Set the root cell at the terminally exhausted.
Right.
An interesting thought. I mean, from the perspective of the computational method, I wouldn't expect as long as you still settle on a linear trajectory, the direction of the gene expression patterns and things like that should all be broadly in the same direction. This is not a purely 100% data-driven approach. So there are certain choices you make in the analysis that are informed by, prior knowledge of biology, which, seeing as this is not a solved problem, some of the existing knowledge about the biology may be incorrect or at the very least incomplete. So that can then, as you use that to steer your analysis, that can then, potentially throw you off course. But yeah, I was able to identify this same kind of linear trajectory from these two pretty clearly identifiable states across 4 different data sets. And when I did differential gene expression analyses between the progenitor and terminally exhausted groups across all 4 data sets, saw a huge amount of overlap in kind of the genes that were up or down in each group. So that doesn't mean that I have 100% confidence about exactly what cellular process I'm identifying, but at the very least, I'm pretty sure I'm identifying a similar process in all of these different data sets. Can you just remind me the last part of your question? Or did that answer it?
It answers it, but I think the challenge I have for you is to go back and think about the fact that trajectory is an assumption that we make based on prior knowledge.
Right.
And that trajectory can be wrong, just like
Right.
prior knowledge can be wrong.
Right.
And if you change trajectory, will you find some truth in the data that has been replicated in prior work that helps kind of untangle the confusion that
Sure.
lies in the current analysis?
I think the next step there, which I have not done comprehensively, but could certainly add, would be to do more of a sensitivity analysis in the selection of the root cells when setting up the trajectory, because that is the most manually user chosen and previous biological knowledge informed step of setting up this trajectory. So I could certainly try. I'm not sure how useful it would be to just randomly pick a bunch of, but I could kind of look at the general patterns and maybe select a number of different root cells and change the direction and see if I uncover something new there. So that's certainly something that I could add to try to get more clarity there.
Are all of those root cells from the same sex patient?
Oh, that's a
You
good question.
use from each of these, of course you get that from me.
Always
but-
the androgen angle.
No,
Uh,
but,
Yeah,
right?
it's a good question. Yeah.
Probably not.
So the single cell data often doesn't have as comprehensive of metadata labeling as the bulk data. So I know for at least for the basal cell carcinoma data set, for some of the data sets, there is patient sex information, but it's not enough to do it comprehensively. But that's certainly anytime you have clear labeling of that, where you can kind of overlay onto your analysis, that's certainly an interesting angle to take. But yeah, I'm not sure.
Well, you can check on the stem cell signature for each individual cells, like see if they are actually really exhausted or not. For example, stem cell signature.
Sure. Yeah. So the root cell population is chosen with these kind of markers of more stemness and less differentiation. So I think that just thinking biologically, that seems like a reasonable place to see the population. Not that de-differentiation can't happen, but yeah, another thing that this would be a much more extensive investigation, but there's been a lot of work using single cell attack-seq and kind of looking at chromatin structure and kind of these epigenetic marks that kind of enforce this progressive trajectory. There's certainly a huge amount more work that could be done bringing in some of these other, not just single cell RNA-seq, but single cell attack-seq, other different data types to try to get a clear sense of what's going on from a chromatin perspective. And that might help with the directionality a little bit. But yes, it's definitely a limitation of this work is that it is informed by previous findings at that one step enough that any limitations in those previous findings, some have the potential to propagate through. Yeah.
So we have a question from online. Nate Evans asks, can RNA velocity help guide the choice of root cell?
That's a really interesting question. The challenge with RNA velocity is a great method. The problem is, at least that comes up for me anytime I think about kind of these kind of time-directed methods, is if the time scales are a different order of magnitude, then I'm not totally sure how effective that would be. I mean, if you have T cells that are being based on my knowledge of immunology, if you get a viral infection, you fight it off in 2 weeks, you're not getting exhausted T cells from that. So it's not, it probably takes, I don't know, on the order of months or something. I don't have the, numbers in front of me, but I would assume it probably takes longer for the cells to be pushed along this trajectory. With RNA velocity, you're kind of looking at something at the level of, spliced versus unspliced RNA, kind of molecular processes that are happening at probably several orders of magnitude shorter time scale than that. So it might be that might be a challenge, but it certainly would be interesting to take a look.
Okay, if we have time, I have one more question. Kind of a comment and just like thinking about this more too, but do you think that for your data sets for the lung, if they're taking, are they taking core biopsies, or do you know what the sample is that they're resecting and like what the location is? Do you have that metadata?
I don't off the top of my head.
Okay.
They are, yeah, these are all samples from tumor tissue, except the HIV data, which is peripheral blood, and you know the tissue context has the potential to change the expression profiles quite a bit. But
Yeah, I was just thinking while Amy's talking too, like it matters too, like the location of where you're at
I
in the
mean, kind of
tumor,
on the margin versus in the center.
Yeah, and so
or-
I wonder if the results that you're seeing are being driven on by like where the location was sampled in the tumor too, right? Like you're maybe towards the edge of the tumor where it's more invasive. If you were to sample in that area, maybe the T cell pattern would change actually. Like maybe your responders would actually have the T cell exhausted low, depending on the part of the tumor that you're looking at. And I don't know if data sets out there exist where they have that metadata of like exact location. But if it does, or if there's spatial transcriptomic data, maybe you can go back and see if your data matches with those.
Yeah, that's a really good point, and kind of a related challenge to that is, my understanding at this point is it's still somewhat of an open question in the field if for the patients who do respond, those CD8 T cells are cells within the tumor that are being modified, or if they're migrating in from elsewhere, and if it's maybe some of both. So that's and bearing in mind all of this data is pre-immunotherapy. So this is characterizing the state of the immune microenvironment prior to these treatments. And to your point, if the sample is taken from the center of the lung tumor and the effect that we're looking for is primarily from CD8 T cells that are recruited from elsewhere, then those are populations that we might not be picking up on to the same degree in these data. There will be data you could collect that would give you a clearer picture, but that's the picture is incomplete because of that.
Thank you.
Any more questions? Fine. So thank you, Chris. It's a great talk.
Thank you. Thanks everyone for coming.